Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 5, Fossils, Dating, and Ancient Environments. In this module, we explore how fossils form, how we date them, and how we reconstruct the ecosystems of the past. These are the tools that allow us to piece together life's deep history, our only way of seeing into the millions of years that came before us. First, let's revisit what a fossil really is. It's not just a bone turned to stone, it's a trace of life transformed by time, mineralized remains, footprints, or even pollen trapped in sediment. Each fossil is a fragment of a much larger story, and taphonomy, the study of how things become fossils, teaches us how that story survives the passage of time. Think about this. Most organisms that ever lived left no trace at all. The fossils we have are the rare survivors of a long geological lottery. Next, we'll look at how we determine their age. Dating methods are the calendar of deep time, whether through layers of rock, chemical decay, or isotopic signatures. These allow us to place fossils within the grand timeline of Earth's history and trace the sequence of evolutionary change. Finally, we'll explore how scientists reconstruct ancient environments, how isotope chemistry, sediments, and animal remains tell us about past climates and landscapes. You might wonder why this matters. It's because understanding how species, including our ancestors, adapted to changing environments helps us make sense of our own relationship with a planet that's still evolving. In this section, we examine what fossils are and how taphonomic processes shape the fossil record. Let's look at what fossils actually are, because understanding this gives us the foundation for everything else in paleoanthropology. Let's revisit a simple idea. A fossil is not just a bone that got old. It's the preserved evidence of past life, and that preservation is rare. Most organisms disappear without leaving a trace, so when something does enter the fossil record, it means it passed through a series of very specific conditions. Think about how quickly a carcass decomposes on the surface today: scavengers, bacteria, weathering. For a fossil to form, all of those destructive forces must be interrupted or slowed down long enough for the remains to be buried and eventually mineralized. When we say fossil, we're not only talking about skeletons. Fossils can be impressions, footprints, burrows, even chemical signatures of ancient organisms. But in human evolution, most of what we work with are bones and teeth, especially teeth. And you might wonder why teeth show up so often. It's because enamel is the hardest tissue in the vertebrate body. It resists decay, survives transport in water, and withstands geological pressure far better than bone. So the fossil record naturally favors what's most durable, not necessarily what was most common in life. Let's think about the process a bit more. Once buried, the original organic materials of bone are gradually replaced by minerals seeping in from groundwater. This slow transformation turns once living tissue into stone while preserving its shape. In some cases, even the internal spaces of bones or the impressions of soft tissues can be preserved. That means fossils are not just objects, they are geological replicas of biological structures. You might wonder why scientists treat fossils with such caution. It's because each fossil is both a clue and a distortion. It tells us something real about ancient anatomy or behavior, but it also reflects the environmental conditions that allowed it to survive in the first place. This makes fossils incredibly valuable and inherently incomplete at the same time. So as we move forward, keep in mind that every fossil we study represents a small victory over time, decay, and geological forces. Each one gives us a glimpse into worlds long gone, and together they allow us to reconstruct the deep history of life, including our own evolutionary story. Let's revisit what we mean by taphonomy. At its core, taphonomy is about everything that happens to an organism after death and before we discover it millions of years later. It's the story behind the fossil, the sequence of events that determines whether remains are destroyed, scattered, altered, or ultimately preserved. And this is important because the fossil record isn't a perfect reflection of ancient life. It's shaped and filtered by these taphonomic processes. So to read fossils well, we have to understand what shaped them. Let's look at the very beginning of that process. When an organism dies, the clock starts ticking immediately. Soft tissues break down quickly, scavengers arrive, insects feed, microbes accelerate decay. Unless something intervenes early, like rapid burial, those remains will simply vanish. Think about a carcass on the savanna today. Within days, it's reduced to scattered bones, and within weeks, even those bones may crack or erode. The same forces operated in the past, which explains why fossilization is such an exceptional event. Burial is essential, but not just any burial. The environment has to protect the remains from oxygen, sunlight, and disturbance. River floodplains can bury bones in fresh sediment. Volcanic eruptions can entomb entire landscapes in ash. Caves can trap remains and shield them from weathering. Each setting creates a different taphonomic signature. A bone buried gently in fine silt will look very different from one transported by a fast-moving river. You might wonder why these physical details matter. It's because they influence how we interpret the fossil. A scattered set of bones might reflect predation, or it might simply reflect water transport. Without understanding the taphonomic context, we risk telling the wrong evolutionary story. As burial progresses, groundwater becomes a key player. Over long periods, minerals carried in water begin replacing the organic components of bone. This slow exchange, called permineralization, turns bone into stone. It preserves the shape, the internal structure, and sometimes microscopic features. Teeth, with their thick enamel, withstand this process especially well, which is why they dominate primate and hominin fossil assemblages. They survive crushing pressure, chemical alteration, and transport better than almost any other skeletal element. Let's think about what can go wrong along the way, because most of the time things do. Bones can be trampled, cracked by heat, bleached by the sun, or eroded by wind. Carnivores might chew them, leaving characteristic marks. Water can sort bones by size or density, carrying some away and leaving others behind. Even after burial, geological processes can distort fossils. Rocks shift, sediments compact, heat alters their chemical composition. The fossil that reaches a paleoanthropologist's hands might be only a partial, weathered, or deformed version of the original organism. Now, this raises a question. How do scientists reconstruct ancient behavior or ecology from such incomplete evidence? The answer lies in carefully reading taphonomic clues. Breakage patterns can suggest whether bones were cracked by carnivores or by geological pressure. Patterns of weathering can indicate how long remains lay exposed before burial. The type of sediment encasing the fossil can reveal whether the environment was a lake margin, a riverbank, or an open woodland. Each clue narrows down the possibilities, helping us move from scattered remains to meaningful interpretations. You might wonder why taphonomy receives so much emphasis in paleoanthropology compared to other fields. It's because hominin fossils are incredibly rare, and every specimen carries disproportionate weight. A single bone can shift debates about locomotion, diet, or evolutionary relationships. But if we misunderstand the taphonomic processes that altered that bone, our interpretations risk being skewed. Recognizing this helps students appreciate why paleoanthropologists spend so much time studying sediments, breakage, and deposition, not just bones themselves. As we move forward, keep in mind that taphonomy is not merely background information. It's the lens through which fossils become meaningful. By understanding how remains move from the living world into the geological record, we gain the tools to read fossils accurately and responsibly. It allows us to see not just what survived, but what was lost, and why that matters for reconstructing our evolutionary past. Let's revisit what we mean by reconstruction and interpretation, because these steps are where fossils stop being isolated objects and start becoming evidence. When paleoanthropologists talk about reconstructing the past, they're not just imagining what an organism looked like. They're piecing together anatomy, behavior, environment, and context to build a coherent picture from fragmentary clues. And the key word there is context. A fossil on its own can tell us something, but a fossil in its geological and ecological setting can tell us much more. Let's look at how this works in practice. Imagine finding a hominin femur in a cave. Before we interpret how the individual moved or lived, we have to ask basic questions. How did this bone get here? Was it dragged in by a predator? Was it washed in by running water? Did the individual actually die in this cave? Each possibility leads to a different reconstruction. This is why taphonomy and reconstruction go hand in hand. The more we understand about how remains were deposited, the more confident we can be in our interpretations. Now, this part is important. Reconstruction isn't only about physical characteristics. It also includes recreating the environment the organism lived in. Sediments, pollen, animal bones, and isotopic signatures all help us infer whether a location was a forest, a grassland, or something in between. When we combine this environmental information with anatomy, we get a richer picture of behavior. Long legs and curved fingers mean something different in a dense forest than in an open savanna, and the surrounding evidence helps clarify which scenario makes sense. Think about this. Even a beautifully preserved fossil doesn't speak for itself. Scientists must infer what muscles attached where, how joints moved, and how teeth interacted. They often rely on comparisons with modern primates, biomechanical modeling, and sometimes even 3D reconstruction. But these are interpretations, not direct observations, and that's why multiple hypotheses may coexist until further evidence narrows the field. You might wonder why paleoartists, those who create scientific illustrations, are included in discussions of reconstruction. It's because visualizing ancient organisms forces researchers to confront the gaps in their knowledge. When you try to draw or model an extinct species, you realize quickly which features are known from fossils and which parts are inferred. This process helps refine scientific interpretations and communicate them more clearly. As we wrap up this section, remember that reconstruction is both creative and constrained. It's grounded in evidence but open to revision. Every fossil discovery challenges us to test what we think we know and sometimes rethink entire evolutionary scenarios. The goal isn't to produce a single definitive picture, but to move steadily toward a more accurate understanding of ancient life, one fossil, one context, and one careful interpretation at a time. First, let's revisit what we set out to understand in this section. Fossils are not simply remnants of the past. They are the results of an extraordinary sequence of events that allowed life to cross the threshold into geological time. When we talk about fossils and taphonomy, we're really talking about how those stories survive, how bones, teeth, and traces endure long enough for us to find them, and more importantly, interpret them. Let's look at what this means for our work as biological anthropologists. Every fossil you encounter is shaped as much by its post-mortem journey as by the organism it once belonged to. Weathering, scavengers, sediment, water flow, all of these act like editors of a manuscript, altering what remains and what disappears. You might wonder why this matters. It matters because our interpretations depend on recognizing which parts of the story are original and which parts have been changed along the way. Now, this part is important. Taphonomy doesn't just tell us what survived, it tells us how to think critically about what didn't. It teaches us to be cautious, to read fossils not as perfect snapshots, but as filtered records shaped by time and environment. That awareness keeps our reconstructions grounded and prevents us from drawing conclusions that the evidence can't support. As we move to the next section, keep this mindset with you. Understanding fossils begins with understanding their journey. And that journey is what allows us to see the ancient world with clarity, humility, and scientific imagination. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.